Welcome to our second podcast on muscle, and what we will accomplish in this unit is talking about cardiac and smooth muscle and those specific things to look for in identifying these two types of muscle. So our objectives, we're going to compare and contrast all the cellular characteristics of cardiac and smooth muscle. We are going to compare and contrast the cellular characteristics of cardiac and smooth muscle. We're going to define and describe the intercalated disc. Now remember, these are those junctional complexes between cardiac muscle cells with a transverse and lateral portion, so we'll need to understand those locational differences and the molecular differences we find within each one of those. We're also going to understand the role of dense bodies in the contraction of smooth muscle, and then we're going to list the locations of smooth muscle throughout the body. So cardiac muscle first, it is very similar to what we studied in Unit 1 with striated or skeletal muscle with some differences that we'll be talking about. Cardiac muscle is only found in the heart and in some cases in the proximal portions of the pulmonary veins, very close to the cardiac muscle itself. And unlike skeletal muscle, which remember was arranged in large cables, these are going to be arranged in sheets or lamina. They're going to be interconnected three-dimensionally, much in the way smooth muscle is that we'll talk about next. Cardiac muscle is striated, which means we will have sarcomeres. We will have the same A and I banding pattern as we saw in skeletal muscle. But cardiac muscle is involuntary. We cannot control our heart rate. Now, true, our brain is going to be functioning to increase or decrease our heart rate, but our cardiac muscles have an inherent rhythmicity. They are going to contract spontaneously with no nervous system input whatsoever. So contracting is an intrinsic result of the heart muscle itself, but the rate of that contraction is controlled or augmented or decreased by our central nervous system. Now, these cells are cylindrical, but the way that we have the lamina formation versus just the cables that we saw in our skeletal muscle is that these cylindrical cells branch. So we're going to sort of interweave or knit the cells into these sheets, much like you knit yarn together to make sweaters and other sorts of clothing. Now, another identifying characteristic to distinguish cardiac from skeletal is that for each cell there is a single centrally located nucleus. Now, recall in skeletal muscle we have peripheral nuclei and multiple nuclei per skeletal muscle cell. Here in cardiac, it's one nucleus, and it's centrally located. Certainly we'll be able to see the intercalated discs, and there are vastly more mitochondria in cardiac muscle than we see in skeletal muscle. Skeletal muscle can endure certain periods of anaerobic respiration. Cardiac muscle is absolutely geared for only aerobic respiration. And to assist that, we have a vast supply of myoglobin present in cardiac muscle. So here's our first view of cardiac muscle, and what we're going to see, we can see essentially, even at this low magnification, the striated pattern of these muscles. You can see it really well here. But you see how we can't just distinguish our long cables. They're just broken patterns, and you can see how they're interwoven and sort of flowing through here. You can also see multiple round nuclei, and these are in the center of these cardiac muscle cells. And then these dark lines in this particular image, these are intercalated discs joining these different cardiac muscle cells to their neighbors. Now in cross-section, we're still going to be looking at essentially the endomysium that we'll see around the cardiac muscle cells, but when we find the nucleus, we're going to see it centrally located and no longer on the periphery like we saw with skeletal muscle. Here's a central nucleus. We're probably in cross-section going to be much more likely to confuse cardiac muscle with smooth muscle because it will have the same appearance of that centrally located nucleus. Now, just a refresher from what we studied in physiology, the intercalated disc, remember, it's the stair step or the lightning bolt that goes between. It's essentially going to be in the place of the Z-line. And we're going to have a transverse portion and we're going to have a lateral portion. The transverse portion, this is going to be like the rise of that stair step that you'd stump your toe against. This is where we're going to have fascia adherens and desmosomes. This is for strength of attachment. However, the lateral portion, or the run where you step your foot on that step, we're going to have gap junctions, and that's going to tie these cells together into a functional syncytium, the cytoplasm of one cell continuous with another. So when one cell depolarizes, so will every cell that it's connected to via the gap junctions. Here we have an illustration sort in 2 dimension. First, we can see here that here we have an illustration of heart in 2 dimensions, and first we're going to see this is the transverse portion, or the rise. Here we have our desmosomes, fascia adherens resisting the force in this direction. And then here we have the longitudinal portion, or the run of our stair-step intercalated disc, and this is where we have the gap junctions connecting the cytoplasm of this cell to the cytoplasm of this cell. Now, a lot of times we think 2-dimensionally as such, so I really like this illustration so you can see the 3-dimensional nature of this. We have drastically increased the surface area for these adhesion points, for the strength of attachment, for the communication between the cells, much like you would in woodworking when you make a dovetail joint. Now again, in these TEMs and light levels, you can't appreciate that 3-dimensional nature, so that's why I really liked that illustration before. But here again, we see our intercalated disc, very dark line, and you see how it's running in the area where we would expect to see another Z line. See, there's a Z line, there's a Z line, and right in the middle of this A, we have an intercalated disc. But they don't perfectly line up. And that's another indication that we're looking at cardiac muscle, because in skeletal muscle, most of these A and I bands are going to line up almost perfectly across all the different cells that we have. Again, in light level, we can see our striated pattern running in this direction. We can see our nuclei are not just peripheral. These are probably connective tissue fibroblasts here. But then we see these dark lines just running along the lines parallel with our sarcomeres. And those are going to be our intercalated discs. Now, as we look at our cardiac muscle, when we look at the sarcoplasmic reticulum T-tubules, remember in skeletal muscle we had triads. We had 2 sarcoplasmic reticula, or 2 terminal cisternae per T-tubule. If you're looking at an electron micrograph, you're only going to see 1 terminal cisternae and 1 T-tubule. So the sarcoplasmic reticulum is smaller. But the T-tubules in this case are 2.5 times larger in diameter than we see in skeletal muscle. So that sort of pushes aside that other terminal cisternae that we would see. So just the dyads is what we're going to see at the EM level as far as looking at our T-tubules and terminal cisternae. Now remember, we are going to have this connective tissue around our cardiac muscle. The endomysium is essentially going to be our external lamina, similar in many regards in composition, almost identical to the basement membrane. So here we have our illustration. Again, we have our very large T-tubules that we're going to be looking for. We have our terminal cisternae as these connection points with our sarcoplasmic reticulum. And you can see how these terminal cisternae are almost going to act like feet, but they're somewhat offset. You can see this one would be in this plane. Here we have another in this plane. So if you're doing a cross section through that T-tubule, it's going to be rare, if at all, you would ever get a triad. Maybe if you did a tangential section, but certainly you would realize that by the shape of that T-tubule. So again, here we have our large diameter T-tubule. There's that terminal cisternae portion of our sarcoplasmic reticulum. Here we have some more T-tubule, very large diameter, and then portions of our sarcoplasmic reticulum. Huge mitochondria. They're not going to be this large in skeletal muscle. Now, within our cardiac muscle cells, thinking back to physiology, remember, we're going to have our sodium channels, and in the sodium channels, we're going to have the fast sodium channel, and we're going to have the slow sodium channel. Those slow channels and fast channels, those are going to allow for the rapid speed of the action potential, but also it's going to allow for the cardiac muscle for a longer lasting contraction. And so in this case, we are going to allow those cardiac muscles to contract rapidly when they need to. However, they're going to be able to have a much more consistent contraction for a longer duration to make sure we're squeezing every last little bit of that blood out of the heart. Again, going back to physiology, just want to mention here and remind you about atrial natriuretic factor or atrial natriuretic peptide. This is going to be expressed by the atrial muscle when blood pressure is too high. This is going to be an intrinsic response. The atrial natriuretic factor is going to feed back through various pathways, as you can see in our illustration, and it's going to result in those things that are going to lead to the production of more urine. It's going to release to the drop in blood volume and that decrease in blood pressure. Now, smooth muscle, again, fairly easy to identify, but we're not going to see any striations. There are no sarcomeres that we're readily going to see. We don't have any T-tubules, and these cells, for all intents and purposes, are going to look a lot like fibroblasts when you see these nuclei in their field. And in cross-section, these are going to appear similar to what we saw with cardiac muscle, but we're going to walk through some of those differences that we'll be able to detect in just the next couple of slides. Now, these are also involuntary, like we saw with our cardiac muscle. They have a centrally located nucleus, fairly oval in shape. And the myofilaments, the thin actin and the thick myosin, they're going to be attached not necessarily at Z lines per se, but they're going to be attached to the sarcolemma in a three-dimensional pattern. And these attachment plaques in the sarcolemma are called dense bodies. And so when the smooth muscle contracts, it's going to pull in 3 dimensions, so that cell is going to take up less volume in space. And when you arrange these into sheets or ribbons, especially those that surround hollow organs, you're going to decrease the diameter or decrease the distance from the ends of these sheets of smooth muscle. Here we have 2 illustrations of smooth muscle in different planes of section. And we're going to see this. This is not uncommon for our alimentary canal. And so in the image that you see, or the section that you see on the left, these are supposed to be longitudinally running smooth muscle. But what you've actually done is you have cut these smooth muscle cells in cross-section. And so what you're looking at are the cut ends of the smooth muscle and the centrally located nucleus. We're going to see these are some ganglia of our enteric nervous system. And then in this case, we have circumferential smooth muscle. So what we're looking at are the longitudinally running smooth muscle. And again, this is going to look a little bit like your dense, regular connective tissue. It's very regular, very parallel, but you're not going to see nearly as many nuclei in ligament and tendon. And certainly these nuclei are a little bit fatter, I think, than what we would see with fibroblastic nuclei. Let's look at another one. Again, showing longitudinal smooth muscle, nice elongated oval-shaped nucleus. And here we've got cross-sections and centrally located nuclei in these cross-sections. Now, in many instances, if you're looking at a high magnification view like this, might be a challenge to pick out those characteristics that we're looking at for smooth muscle. And so we're going to have to pay attention to where we are in the body and the tissues surrounding our smooth muscle to help us know exactly what we're looking at and where we are. So where do we have smooth muscle? Well, certainly the walls of our hollow viscera, which are going to be our digestive tract. Certainly we have these in our respiratory tract. Walls of blood vessels. The larger our ducts of our glands are going to be, the more we're going to need smooth muscle around to create somewhat of a peristaltic action to help express those products. And then lastly, when we look in the dermis of the skin, we have small bundles of muscle. These are the erector pili muscles that cause your hair to stand on end. Those are going to be smooth muscle. Now, their arrangement into functional groups, we have multi-unit smooth muscle and unitary. So multi-unit, each cell can contract independently of one another. They will still be innervated by the same axon. And remember from physiology that you have these varicosities along the length of the axon. It's not one just terminal bouton at the end. But as these varicosities pass through the smooth muscle, it will allow for the neurotransmitters to be released and for those chemicals to be picked up by chemically gated channels. And so that's called synapses en passant, or in passing. Now the unitary type, this is going to be where all the cells function as a syncytium, function as a unit because of the gap junctions between these cells, and these are going to be indicative of what we're going to see in our digestive tract. These large sheets and ribbons of smooth muscle, the sphincters, for instance, are going to work together to help move our food through our digestive tract. Now, lastly, a little bit that we're going to see with smooth muscle, we are going to see caveolae. I know this is down here at the bottom. The rest of this we're pretty familiar with. No troponin. We do have some tropomyosin. We're not going to be functioning the same way with calcium because remember, as calcium comes in, calcium is going to bind and stimulate a cascade event that is going to lead to the phosphorylation of myosin light chain kinase. That activation of that kinase through the calmodulin calcium pathway is going to lead to the phosphorylation of the myosin light chain. That myosin light chain, when it becomes phosphorylated, it opens up so we can have that contraction. In the caveolae, this is where we're going to store our calcium in these little bubbles located beneath the plasma membrane. And so we're not going to have sarcoplasmic reticulum, but we're going to have these little bubbles, or caveolae. We will look at those when we get to lab. We're not going to see them at the light level. Certainly we'll be looking for the caveolae primarily at the M level.